Welcome to the Functional Medicine Doc Talk podcast. I'm your host, Dr. Kurt Woeller, talking with experts in functional and integrative health and medicine, discussing critical information for improving your health and wellness to help you live a long, full life. Let's get to it. Hi, and welcome to the Functional Medicine Doc Talk podcast. I'm your host, Dr. Kurt Woeller. And I'm joined today by a very special guest, somebody I've known for over 25 years, going way back to the late 90s when I first saw Dr. Bill Shaw, a PhD of Great Plains Laboratory, speak at a Defeat Autism Now conference in San Diego. So we've worked for many years alongside each other, lecturing at many conferences. I've used the Great Plains Laboratory testing for many years and have learned and continue to learn so much from Dr. Shaw. So I'm really excited about this conversation. So Bill, welcome to the program.
Oh, thank you, Kurt. Great pleasure to be here.
So we're going to get into organic acid testing. You have a new paper that recently came out looking at palmitic acid, HPHPA, secondary, elevation from HPHPA and other types of phenylpropionic acid derivatives. But before we get into that, give the audience a little background on yourself. How did you get into this work? You ran Great Plains Laboratory for 25 years or so. So we'll talk about that and get into this important topic.
So really, the organic acid test was something I had wanted from the very beginning of my career. Even when I just graduated with my PhD, that was a major goal of mine. The idea was, mass spectrometry at that time was just becoming accessible to some degree. And I thought using this technology, you'd be able to test every single biochemical that's inside a person by testing their urine. And so that was really my goal from almost from day one. It was kind of ridiculous in a way, but the very first day I started my first job, which was at CDC, my first question was, if I get the Nobel Prize, do I have to give the money to the government? Or can I keep it? And the HR guy said, nobody's ever asked us that question before. That's going to take a while to find out.
That's a good question.
But I had started preparing for the organic acid test almost immediately. I had started setting it up in the lab at CDC, looking at the technology. And at that time, the technology available was just gas chromatography. So the thing is, I got my hands wet. I found out what the challenges were, and realized that mass spectrometry would have to be used, and so that was the way to go, and high-speed computers. And at that time, there was not the technology to connect the computer to the mass spectrometer. That came out later, and so from CDC, I wanted to learn mass spectrometry, so I got a job in drug testing, which was a place where mass spectrometry attached to computers were starting being used, and so I learned all about that. Gained hands-on experience with that technology, and so really, my setting up Great Plains, there was a lot of background stuff that had to be done, to prepare myself for the challenge of setting up a laboratory that was going to look at not only genetic disease but all disease, to find out what the underlying causes of all disease, with the idea that using a urine sample, you would be able to do that. And to some extent, I accomplished that at Great Plains. Not every disease, but a lot of got viewpoints into the underlying causes of a lot of diseases. And some of the stuff, of course, it just the information, the research really still hasn't gotten out there. It takes a tremendous period of time for the learning process to go on and for this kind of information to get into the medical education and stuff like that. So,
well, it's a complicated test. I mean, I taught the organic acid test seminar now for many years, and there's so much to learn. I'm still learning. And when I first started using the OAT test, I remember having to reach out to you because I was so overwhelmed by the amount of information that we got. And so real quickly, by the way, why is mass spec so much more effective? And exactly what is mass spec for people who have never heard that? Why is that the preferred method for this type of testing?
Yeah, it is because it is the most specific way of identifying a compound. So other things measure the ultraviolet light absorption or fluorescence or some other characteristic of the molecule, but almost all those other technologies have interference. So mass spectrometry is one of the few technologies available where there's almost no interference. You can't say 100%, but like 99.9%. So even if you have chemicals that are coming out on the same place on what is called a column that separates molecules, you can still determine that there's 1, 2, 3, 4, or maybe even 5 substances in a single peak. It's not a single substance. So it's highly specific. It's the most specific thing. So, at the sporting events or the Olympics where they check for steroids, that's the technology that's used.
Okay.
Because it is close to 100% accurate.
So you wrote a paper that I first looked at many years ago that was looking at some autistic boys who had very high levels of fungal markers. And then you came, I don't know if it was before or after, you had another article looking at Clostridia and specific compounds secondary to the existence of Clostridia. And so we could talk for hours just about the fungal component, but I want to really keep our conversation here focused on the Clostridia component to get to your new paper. So when you came out with that information, we're looking at Clostridia. Most people think of Clostridia as Clostridia difficile, a bacteria that's primarily going to generate bowel disease of some form or another. But you discovered that certain Clostridia were producing other compounds that were having negative effects sort of outside the digestive system. Can you explain a little bit about that?
Yeah. So in preparation for the mass spectrometry organic acid test that I set up at the Children's Hospital in Kansas City, I did a tremendous amount of research on all the genetic diseases. I talked to other laboratories that performed organic acids. And then I decided I was going to do more than the standard. So I added substances that were present as food additives and vitamins. And so some of the things were not offered and still some of the things are still not offered by any other laboratory in the world. And so so I wanted to have every single thing that could possibly be tested included in the test. So to open the scientific eyes, so to speak, and so when I had done it, I was confident that I'd be able to solve new diseases, and so I went to there was a children's psychiatric hospital in Kansas City, and I made an appointment with the medical director and told him about my new test and that I had something I thought would work very well for cases where they really didn't know what was causing the illness. And he said, I think I've got the perfect case for you. I've got a kid who's got depression and attention deficit and oppositional defiant disorder. The kind of kid, if you tell him to turn left, he goes right. If you say stop, he goes. So he does the exact opposite of what you ask him to do. He won't cooperate in any way. And he sent a sample in, and I saw what turned out to later to be identified as HPHPA. And so I thought, well, that was interesting. But the kicker was a couple weeks later, he deteriorated and had to be hospitalized. And they sent a second sample. And this sample had a gigantic peak. It was the largest peak I had ever seen for any compound, like larger than phenylketones and the genetic disease phenylketonuria. So it was higher than most of the genetic diseases where you have gigantic amounts of the substance. And so at first I thought maybe I had found a new genetic disease. But the fact, though, that this compound was so gigantic, it blotted out all the other compounds. You couldn't even see them. It was like, so I mean, a common value for some organic acids is like 20. Well, this was 10,000. So it was absolutely gigantic. So immediately I knew that I had found something important. The fact that it was still slightly elevated when the kid was, an outpatient, but when he was hospitalized, it reached a gigantic level.
Did you know what HPHP was at the time? I mean, I knew you knew how to identify it from a chemical structure standpoint, but did you understand the significance of it as far as what it did chemically?
Well, I just knew that it must be something extremely important, and that and so my first concern was that maybe it was a drug, but anticipating this, I had actually developed a library of all like the common psychiatric drugs like phenothiazines and other psychiatric drugs, because I thought maybe it's just he's on drugs, and so this is a huge drug metabolite, but it wasn't. So I was able to find that it was not any of the common drug metabolites, and also it wasn't any of the genetic diseases. So it either had to be something new, and I talked to the leading people in the field of organic acids, people that were known worldwide for organic acid testing, and they said either I have no idea what this is when I showed them the mass spectrum, or either I have no idea what it is, or it's something from the microbiome, which I don't think that name was even used. I just said,
wow. So they acknowledge that, and that's interesting. Yeah.
But they said it's not an abnormal. It's not due to any genetic disease. So just forget it. It's not worth pursuing. So luckily, I completely ignored that and started making that the focus of my research from then on. And
hey, by the way, do you remember what happened with that child from that first test to the second test? Did they get put on antibiotics of some sort, or you remember any specifics?
I do not. That would, I mean, that's a great question, but at that time, I did not. So at that point, I just had to say, report back that the child was negative for all organic acid genetic abnormalities. He did not have any of those.
So where did you make the connection then between HPHPA coming from, which you eventually found was coming from Clostridia, certain types of Clostridia bacteria? As we mentioned, most people think of Clostridia as Clostridia difficile, and that produces its own compounds, but the HPHPA comes from different types of Clostridia. When did you start making the connection of, hey, this is something really bad, and it appears to be doing X, Y, and Z in a certain select types of patients? and
Yeah.
here's the mechanism for it. When did that start to evolve?
So, with my, I had developed some knowledge of in mass spectrometry interpretation by this time, and so I was able to find, I realized it was a phenol, which means it has a benzene ring, a 6-carbon ring, and a OH, a hydroxyl group attached to it, and so which is also a structure that's similar to the neurotransmitters. So I thought, well, this could be very significant because it's interfering with somehow with neurotransmitters. So I had a rough idea of what the structure was at that time. And then I was noticing that it was also present in a lot of the kids with autism, and eventually found it was significantly higher in the autism population. Doing about something like 60 kids with autism who had high values, and it wasn't high in normal children. So that became significant. And then later on, I did a test of, since I was looking at the effect of yeast and fungus, and so there was a study. It was a formal research study that was approved by the board at Children's Mercy, and we gathered 23 kids with autism and put them on nystatin, and a lot of the compounds that I thought were due to yeast or fungus indeed were, and the values of those compounds dropped after the kids were on nystatin, except for the compound, which I now call HPHPA. It actually went up after being on nystatin. So to me, that proved it wasn't a yeast or fungus. It was likely a bacteria. And just about that time, I had a parent who'd sent in her child's result, named Ellen Bolte. And her child had a severe regression after getting the DPT shot. And so she thought that it was perhaps the tetanus in the DPT shot had caused an abnormal reaction, because she looked at what are some of the clinical symptoms of tetanus, and some of them are the person has difficulty talking, some of the same things that are present in autism. So she thought that maybe the tetanus shot was actually contaminated with live tetanus bacteria, that they didn't sterilize it enough, and so her child had developed a kind of a subclinical tetanus. And so she suggested that it may be a tetanus bacteria. And tetanus, of course, is a Clostridia bacteria. So I started looking at Clostridia bacteria, and then eventually I found I knew what like 99% of the structure was. The only thing I didn't know, I knew all the atoms that were in the molecule, but I didn't know exactly how they were arranged. And there were probably about 100 possibilities of how something with all, I could get, like it has so many carbons, so many hydrogens, I could get all that information using advanced mass spectrometry that I had to farm out to a research company. And so now I needed to know how would this relate to Clostridia bacteria? And I was now able to know that it was a compound called a phenylpropionic acid. Well, phenylpropionic acids, I did more research that was in the literature and found that phenylpropionic acid is produced by eight species of Clostridia bacteria. And tetanus is not one of the ones. So I was able to rule out the idea of Ellen Bolte that tetanus was somehow related. But interesting enough, one of the things that was found was the one that causes the food poisoning, botulism, is one of the ones that does produce the phenylpropionic acid. So I was able to figure out, I was very good at metabolic pathways and was able to figure out that the chemical that was in the gigantic amounts in the kid and in kids with autism would be converted to HPHPA. I was able to figure out, using my knowledge of human biochemistry, that the phenylpropionic acid produced by the Clostridia would be converted in the human to HPHPA. And
go ahead. Yeah. I was going to say, before we go down, because we're stepping into the area that I really want to investigate: the phenylpropionic acid, HPHPA, those fatty acid those pathways. Before we do that, I actually have a question for you. So, going way back, when I first did my first organic acid test, you helped me with a child who had pervasive developmental disorder. He had high levels of Candida, the arabinose. We put him on nystatin. He did a gluten casein-free diet, eventually lost his diagnosis. But I remember a particular case years later was a child that had all the characteristics of yeast candida overgrowth: goofy, giddy, silly, inappropriate laughter, high self-stimulatory behavior. And so I said, well, I don't really need to run a test. I already know what this is. So this is candida. So I put him on nystatin. And within 3 days, he went from goofy, giddy, silly, inappropriate laughter to aggression, self-injurious behavior, headbanging. I had some experience at that time seeing that behavior being linked to Clostridia. But in this particular case, I decided not to do the test. I assumed, right? And it was a total Jekyll and Hyde moment. Treated him for the Clostridia, or what I thought was, eventually confirmed it later. So my question to you is, and I've not been able to figure out the mechanism, but we've talked about this. I know you've seen this pattern too, and I'm wondering, in that particular individual, where you went nystatin, the yeast markers dropped in the oat, but the HPHPA goes up. Do you know the mechanism of what's happening? What
I think it's just
competition.
It's fighting for food and space. I think that's really the organisms are in a competitive ecosystem in the GI tract, and so when the yeast compete to keep down the amount of Clostridia, when the yeast are eliminated, the Clostridia just takes off. So, and the same thing, of course, happens if you give antibiotics. So Clostridia is not killed by the common antibiotics. So if you give things like penicillin or and other antibiotics, the things like the common strep and staph and organisms like that may be killed, but Clostridia is not, and so it just goes to town. And the same thing happens with the glyphosate. Glyphosate kills all the beneficial bacteria, but does not kill most of the Clostridia, or all the Clostridia species that have been evaluated so far. They actually proliferate. And to me, this proliferation is one of the large ecological problems in the whole world. It's one of the biggest problems, because continuing to spread glyphosate throughout the whole world is transporting the soil from normal, healthy soil to contaminated soil, so that all the soil we're using for farming is becoming contaminated. Even cattle eating the grass are dying of things like botulism because of the use of glyphosate. So this is it's almost equal to the climate problem on the earth. And so this one, of course, is caused by human technology. So the idea that humans can just change technology at random is just amazing. So it's one of the things like that you don't want to make a gigantic change in our ecosystem without very, very carefully looking at what may happen as a result.
So I'm going to come back to that. And that topic, I know acetaminophen comes into this as well. So real quickly before we move on, I'm going to talk about HPHPA, dopamine beta hydroxylase, how that plays into this, your new paper. So the moral of the story for me through all of this, right, that Jekyll and Hyde scenario with that kid was never skip the oat. Don't ever skip on doing the organic acid test and just assuming you know what it is based on symptoms, because in this particular case, it was a total disaster. So you figure out, you identify HPHPA, you start to realize it's produced by different types of Clostridia, including Clostridia botulinum. And then it's linked to this phenylpropionic acid. So we have imbalance in the microbiome, these overexpressions of these Clostridia bacteria. And let's talk about one of the things I've lectured a lot about, learned it from you, was the inhibition of dopamine beta hydroxylase, is a very important enzyme in the nervous system. And then, of course, we get increases of dopamine and some of the consequences. So let's talk about that. Explain to me how you made that connection, and then let's get into your new paper. So what was the connection between HPHPA and dopamine beta hydroxylase, and what types of patients does that fit into?
Yes. So that was a very important realization that was going on. So the advantage of the organic acid test is it measures the major metabolites of dopamine, which the major metabolite is abbreviated as HVA, homovanillic acid, and the product of dopamine beta hydroxylase, which is VMA, vanillyl amandelic acid. So by looking, in one test, we could see what was happening to the Clostridia compounds, at the same time seeing what was happening to dopamine. And so what I was seeing is that the higher the Clostridia, the higher the dopamine values became. And that in reviewing the literature, I found out the high dopamine was something that had been reported, consistently over a period of 50 years. So it was amazing to me that researchers didn't jump on this. And a matter of fact, some of the people have said, part of this had to do with, the DNA people being at the National Institutes of Health, so an inordinate amount of research was being focused that way, and autism was being ignored because except with looking at the possibility that autism might be genetic. And of course, the latest data show that perhaps 15% at most may be involved with genetic. 85% is non-genetic. So people are hopeful. There's a new NIH director who has a son with autism, and their hope that perhaps there'll be a switch and more focus to resolve autism, which I think is now completely within the reach to nearly completely eliminate.
Yeah. So, we'll see. I'm not sure I'd hold my breath on that, but we'll see. So one of the things I've seen is elevations of dopamine. We know it. We get the metabolites of the quinone metabolites, which have neurotoxic effects. And we generate a lot of reactive oxygen species. You get depletion of neuronal glutathione and then neurodegeneration. So we're talking more than just autism. right? Because you
Yeah.
started.
So it ends up that this HPHPA, once we started expanding the scope of the patient and physicians who use the testing, we found it virtually every neuropsychiatric disease. There was a significant percentage of the patients who had the problem with the Clostridia, and it's not surprising since Clostridia causes elevated dopamine. Elevated dopamine is the target for the neuropsychiatric drugs like haloperidol, one of the first neuropsychiatric drugs, is blocking dopamine receptors. And why did dopamine receptors have to be blocked? Because there's too much. So this is probably one of the most important scientific discoveries of the time, but because of the huge influence that the drug industry has, it's their focus is on having psychiatrists spend more money on producing more expensive drugs and making a higher profit, and so that is. And I understand that, if that's your job, you do your job, you can. But unfortunately, that job is not helping to get at the underlying causes of many neuropsychiatric diseases. And just to give you an example of the ones that I've seen where very high values of the Clostridia were present: ADHD. The depression, both unipolar and bipolar, and Parkinson's disease, seizure disorders, anorexia nervosa. And so you can see, like an anorexia nervosa, if the person has severe Clostridia, the Clostridia is produced from amino acids that are in meat proteins. So the person won't eat meat. It's not because the person is trying to make life hard for the parents or something like that.
Mm.
They realize that anytime they eat meat, more Clostridia is formed that messes with their brain. And so it's a defensive mechanism. And the solution, of course, is to get rid of the Clostridia, and then the person will be able to eat meat.
I've actually found Clostridia in some cases to be associated with, I wouldn't say it's the direct cause of all, of individuals with, postural orthostatic tachycardia, forms of dysautonomia, or at least exacerbating it, and exercise intolerance. I had a case once of a woman who had poor exercise tolerance going back to when she was a teenager. Now, it turned out that she had some genetic variants in the dopamine beta hydroxylase gene and the way that the enzyme was expressed. But on top of that, when she had a Clostridia problem, it just exacerbated the problem. So it's so important. But Bill, I would say that it's not one of the most significant medical discoveries of the past. It's one of the most significant medical discoveries ever, and it still is very, very important even today, and particularly now with the new paper that you've written. And it's a paper that I find extremely important. Once this podcast is posted, I'll make sure to put the link for people to be able to access that paper. So the paper is dealing now with HPHPA, which we know has been along. We've talked about this. You've been talking about this for many years. Of course, the link to dopamine beta hydroxylase issues, excess dopamine, et cetera. But now your research has taken you into a different area in looking at how the elevated HPHPA, maybe you can explain a little bit more about some of the phenylpropionic acid, if there's other organic acids that are sort of similar in their structure, using up coenzyme A, which then affects fatty acid metabolism. And that has a huge impact, we know, from metabolic efficiency, mitochondrial function. But specifically, you're looking at palmitic acid. So tell us about your new paper and some of the new discoveries. Let's go down.
Yeah. So so that was one of the benefits of my selling the company, Great Plains Laboratory, and then becoming an employee, is I had a lot of free time. And a lot of free time is necessary to do all the literature research that's needed to see what is everybody else in the world doing. And I found really, so one of the most useful thing were papers in metabolomics, which means that people look at every single compound that's present in the urine and people with autism and see what are the ones there. And what are the ones there were ones that I already knew. So if several of the labs had found HPHPA was, if a person had positive HPHPA and a few of its metabolites, there was a 98% certainty that person had autism. And so that was extremely important. And also, they had found that another chemical, a really basic biochemical that doesn't seem to be specific at all, was the most important compound for determining how severe autism was. And that was palmitic acid, the chemical that comes from palm oil, so, which is from palm trees. And the reason I was so impressed was because two research groups that were thousands of miles apart, probably who had never spoken to each other, both found that palmitic acid was found in about 90% of children with autism. So one lab was in Italy, and the other lab was in Pakistan. So both of them found this was not found. There was no detectable palmitic acid in normal children, but there was large amounts. And the higher the palmitic acid in the urine, the more severe the autism. So this blew my mind that something commonplace was one of the major factors in autism. As a matter of fact, I recalled that maybe when I first setting up the lab that I had seen palmitic acid, but thought maybe it was an artifact that how could this thing be so. I at that time I didn't know that. I thought fatty acids weren't found in you, which of course in normal people they're not, and so I thought maybe it was a artifact that palmitic acid was present in a child with autism. So now
you also mentioned in your article too that there was nothing to believe that a diet of an autistic individual's diet was higher in palmitic acid compared to other individuals. It wasn't right. They were eating super amounts of it.
No, they weren't. No, they weren't eating high amounts of that at all. So since there was no indication that they were eating high amounts, it had to be that their metabolism was different, and so I tried to put those things together. The fact that HPHPA was one of the most common elevations in autism, and palmitic acid was also, and so I had to go back to one of my talents is be able to develop metabolic pathways for which nobody else had published, to come up with new metabolic pathways. And so that's what I did using the HPHPA, figuring out that since it comes from phenylpropionic acid, and it's well known that phenylpropionic acid had to be broken down by what's called the beta oxidation pathway of fatty acid breakdown. And this was something that had been known for about 130 years, that this is how certain fatty acids are. So phenylpropionic acid, in addition to be something that messed up dopamine, also messed up the breakdown of fatty acids. So it used up one of the key vitamin coenzymes in the body. So to break down phenylpropionic acids, the body was using up almost all its coenzyme A, which is a critical cofactor in all of human metabolism, and not only human, all of animal metabolism, probably going all the way back to even bacteria and yeast metabolism. So all of life depends on this coenzyme, having adequate amounts of coenzyme A. And since having high phenylpropionic acid was going to use this all up, it was going to cause a deficiency of coenzyme A, because all the coenzyme A would be attached to the Clostridia compound, and so it wouldn't be available for doing probably about 100 other biochemical reactions in the body that need coenzyme A, and those include making cholesterol, for example. Kids with autism have deficient cholesterol. Having not enough acetylcholine. It's another thing in which there's evidence that that's deficient. And acetylcholine is important for the brain, but also for the muscle function as well. So in the brain, not having enough, it impairs memory if there's not enough acetyl-CoA. But it's also needed to make new fatty acids. It's needed for breaking down amino acids. It's needed for producing energy and ATP. So it has so many functions. So if the Clostridia is there, it's going to really mess things up. And so the other thing is palmitic acid is needed, or coenzyme A is needed to activate palmitic acid so that the palmitic acid can be broken down or transferred to a key developmental protein called sonic hedgehog, which is named after the video game, the Sonic Hedgehog, which is like a porcupine-looking rodent animal that's blue colored, and so if the coenzyme A is not available, palmitic acid can't be attached and utilized, and if it can't be utilized, it'll be excreted in the urine in excessive amounts. So that's exactly what I think is going on. So the kids with autism who have a defective sonic hedgehog, they will have more sonic hedgehog than normal children, but it's all defective sonic hedgehog.
So Bill, before we move on to that, let me just kind of recap. So
I know it's a lot of stuff.
That's okay. It's a lot of stuff, but it's great, really important. So we've got Clostridia. Okay, so we've got these various Clostridia that we come in contact with, can develop in the microbiome, certain susceptible individuals. We mentioned before that glyphosate can be a component of this, causing a depletion of healthy microbiome bacteria and allowing for an overexpression of these pathogenic bacteria. So we're producing HPHPA, and we're getting a lot of this, and it's sequestering, it's using up a lot of that coenzyme A. Now, coenzyme A comes from B5. So maybe we can come back to that, towards the end. But and we know that coenzyme A is used metabolically through many different areas of the body. So it plays a big role, let's just say, in metabolism and ATP production. And then because of a lack of coenzyme A, we're getting an accumulation of palmitic acid. So palmitic acid can't attach appropriately to enough coenzyme A, so it can't enter its fatty acid metabolism pathway. And then we have an issue with sonic hedgehog. Now, sonic hedgehog is a protein that I learned about from you in relationship that it's very important for neurological development, for digit formation, for limbs. And I think what's important for people to understand about sonic hedgehog, it's not just something that's produced as in an embryo or when you're a young child. It's something that is being produced throughout our life.
Right, for your entire life.
To maintain this normal or these structural components of our body. So we've got high HPHPA, lack of free coenzyme A, too much palmitic acid that can't attach to the coenzyme A, and now we have inactivation or a lack of activation of sonic hedgehog. So you were mentioning in autism, the studies have actually shown elevated sonic hedgehog, but you're stating, well, that's the inactive form, essentially. Is that the idea?
Right. Yeah, that and of course that will be the ultimate proof that the theory is correct. We'll be testing the sonic hedgehog and finding out that it's all sonic hedgehog without palmitic acid attached to it. So it would be just like the feedback loop with thyroid. So the thyroid gland produces T4 in response to the pituitary hormone TSH, thyroid stimulating hormone. And if there is if the thyroid gland doesn't produce T4 because, say, there's a deficiency of iodine, so there's no iodine in the diet, so the thyroid gland can't produce T4, what happens? TSH gets higher and higher and higher. So this is my theory as what must be going on in autism, that the sonic hedgehog is being produced to do the kinds of things it needs to do in order for development to occur. But because palmitic acid is missing, that is one of the reasons that the sonic hedgehog being produced is all defective. Just like, if you had, for example, if TSH was defective and the pituitary was producing it, it wouldn't stimulate the thyroid gland. And so there'd be more and more defective TSH produced because none of it was working. So that, in effect, is what's going on. And the higher the TSH, and in autism, the higher the sonic hedgehog, the more severely abnormal the child is.
Is there any evidence? I'm sorry, I'm adjusting my screen here. I got this weird light coming through my window. But any evidence?
Oh, yeah, I see it.
Yeah, it's just right across my ear. So any evidence that inactive sonic hedgehog is somehow damaging itself? Kind of like, I'm wondering if sonic hedgehog breaks down into some toxic metabolite like dopamine breaks down into quinone. Is there any?
Well, I mean, so this protein, I don't think people know the exact half-life. So even normal sonic hedgehog is going to break down after a period of time, and then the amino acids that make up the protein will be recycled. It might well be toxic, but I think the biggest problem is just that it's non-functional. Right. I think that's the biggest problem. But the future research will determine that. So I see this as an explosive research, or it should be an explosion of research into the function. Almost all of the research today is just looking at its role in cancer. That's almost all the research, and it has a role in cancer as well. So which will be interesting to find out if it can be used as a cancer modulation.
So you mentioned coenzyme and cholesterol. So we've got HPHPA. We have now sequestering of coenzyme A. We've got dysregulation of other biochemical pathways, one of them being cholesterol. We can't connect enough palmitic acid to coenzyme A to enter its metabolism pathway. So we have a buildup of palmitic acid. And then that affects sonic hedgehog. So we have this inactive sonic hedgehog. But then you mentioned cholesterol. So there's another component to activating sonic hedgehog. It's the palmitic acid.
Right. Yeah. And so one end of the sonic hedgehog, and it's called the N-terminal. So every protein has a amino group at one end and a carboxyl group at the free carboxyl at the other end. Every single protein and of every living thing, of every protein. So the palmitic acid attaches to the nitrogen end of the molecule in order to activate it. The cholesterol attaches to the carboxyl end, and the lack of either one of those makes it inactive. And so that is something that has been proven, that if these things are not attached, the molecule does not function correctly. That's something that's not theory, that's a proven fact.
Very interesting. And then I'm curious with regards to perhaps, beyond autism, for example, are there we mentioned cancer as well. Do you know of any other mechanisms of sonic hedgehog that maybe in an adult with Parkinson's or Alzheimer's or some other type of neurological condition where there is some type of imbalance with sonic hedgehog inactivation that could?
Yeah, so you're right. So any of the same things that happen to the person with autism are likely going to happen to any of the people with these neuropsychiatric diseases where the sonic hedgehog is not being activated correctly, where it's not functioning correctly. So it's fascinating. So sonic hedgehog is probably one of the major proteins in evolution. It's probably why, the whales have, I remember as a kid at the museum seeing that whales have little tiny hind feet that are just vestigial. So at one time, the ancestor of the whale had, 4 legs, but now, those legs have been converted to fins, and they've probably been converted by changes in sonic hedgehog. So changes in sonic hedgehog over the thousands of years have probably one of the main the main mechanisms of evolution of life on Earth.
Very, very interesting. Yeah, there's a lot of mechanisms. We were talking earlier. We'll see where the research. This is something I'm looking into. Is palmitic acid is a known activator of some of the toll-like receptors that are part of the innate immune system, and then that sequencing, to transcription activation of NF-kappa B and inflammatory markers. And one of the other things too about that from a neuroinflammatory standpoint in your article, and what's known in autism particularly, is big issues in the cerebellum. And so, the cerebellum is often dysregulated in autism from a proprioceptive standpoint. And I believe it was in your article, you mentioned that the Purkinje cells produce sonic hedgehog
right,
as a means of developing normal structural integrity and connections within the cerebellum. So there's just so much there, Bill, that is already known, but I think it's such incredible research and insights that you've come up with. I've read your paper twice. I'll probably read it again because there's a ton of information in there and a lot of other areas to start thinking about. One of the other things you commented about was acetaminophen. And we have talked a lot about this in the past and some of the toxicity of acetaminophen and the compounds it produces. Tell us a little bit about acetaminophen and its connection to this discussion.
Yeah, so the acetaminophen has several metabolites, and the most toxic of these metabolites is it's a handful to say completely, so I use the abbreviation NAPQI, and I call it NAPQI, just to make it a simple word to remember. And NAPQI is the thing that causes acetaminophen is one of the major causes of liver failure, deaths from liver failure from taking excessive amounts. And the thing that is probably the main factor is the NAPQI. So the NAPQI is a very reactive molecule that reacts with sulfhydryl groups. That's groups that have a sulfur group attached. And what do you think the very end amino acid of sonic hedgehog has this sulfur group? So it is a target for the NAPK. And it's been found that NAPK can react with, to my knowledge, every single protein which it's been studied. It has the ability to react and inactivate those proteins with their and activating their sulfur group. So if it reacts like that, and I think the evidence would be very strong that it would, since it reacts with every other protein, it wouldn't make sense that it would not react with sonic hedgehog. So if it reacted with that, then palmitic acid could no longer be attached. So you would have a dysfunctional molecule in which the molecule had a drug attached to it instead of the what nature intended, which is a palmitic acid.
So what about, which then raises the question about heavy metals? If you think about mercury, lead.
Yes, mercury would be another one. So that's another one. So mercury also has the ability to bind to that sulfur group on the end of. So all of these molecules can now be looked at using mass spectrometry. You could look at sonic hedgehog containing mercury, sonic hedgehog containing NAPK, and also one of the other Clostridia metabolites called cresol also forms a benzoquinone that's very similar to NAPK that'll also react with that end sulfhydryl group on sonic hedgehog.
So so what you're saying then is in an individual who has, and I see this quite a lot, elevated HPHPA and 4-creosol, it's a double whammy effect almost.
That's right.
And then in a different way, but on that sonic hedgehog. Wow.
Yeah. Yeah. So all three things, the mercury, the NAPK from the acetaminophen, and the cresol from the Clostridia, all three of these have the ability to inactivate this sonic hedgehog. So there really is a critical need for this to be dealt with like the way that the government dealt with the COVID vaccine, put a tremendous effort. I mean, business as usual to me is a big problem. It's estimated that if untreated, that the autism is going to cost something like $5 trillion a year for to handle all, because when children are little, the parents are going to, but eventually the parents aren't going to be able to do it anymore, and the children will have to be adults who are going to have to be taken care of by the government, and so a huge amount of governments throughout the world are going to be affected like this unless something is done. So.
It's an absolute disaster. It is. It's an absolute. I totally agree. It's I mean, I've been at this for over 25 years, and you even longer, and it's getting worse. It's getting worse. It's not getting better.
The incidence was in the around 1970. The incidence was about 1 in 10,000. And now it's gone up to, I think the last word was 1 in 36, and in California, 1 in 13 boys. So you're talking about a tremendous increase in the rate of autism. And of course, some people try to claim that it's just increased diagnosis, but other people have made arguments just as convincing that it's real, that it is not. There is some degree a better diagnosis. But if it was all better diagnosis, then you would say there would have been a lot of 70-year-old people who were never diagnosed. If we went to the around, we would find all these undiagnosed autism older adults, but we don't find them because they're not there.
So, Bill, as we wrap this up, I mean, it's a lot of information to take in, but we clearly know that acetaminophen use, for example, right, really not a good idea. Certainly don't want to be preloading kids with acetaminophen prior to their shots and staying away from it as much as possible. Clearly, the glyphosate is an ongoing, problem, which unfortunately I don't see going away anytime soon. And we know that clearly is something that disrupts the gut, disrupts the microbiome. The expression of these Clostridia bacteria, the HPHPA, and now we've got the palmitic acid, the cholesterol, the sonic hedgehog problem. What can people do? And I know I'm not expecting you to have all the answers for this because it's ongoing research and development. We know that we can, deal and treat with the Clostridia and try to improve the microbiome status and eat organic food, stay away from GMO food and those types of things. But when we're talking about fatty acid metabolism, one of the things that often helps is carnitine, and I'm wondering if carnitine would be something that might preserve some of that coenzyme A, that it's not.
That would be an excellent idea. So the carnitine, also pantothenic acid is like available from New Beginnings that would that is part of coenzyme A, and studies have shown that pantothenic acid is commonly deficient in the urine of children with autism. So that seems like a logical thing that could be done. If the children have low cholesterol, supplement with cholesterol. So cholesterol is not always bad. So low cholesterol is just as bad as too high cholesterol. So all of these things need to be done. And probably the most important thing is never use acetaminophen. Use and studies have shown that mild fever actually helps to overcome an illness. So don't even use analgesics if the fever is mild. Don't use it for every instance of fever. Or if you have to use it, you can use ibuprofen instead, which doesn't have the same autism incidence associated with its use. So the studies showed it great. John Hopkins is that ADHD and autism were both significantly elevated if the mothers took acetaminophen in the pregnancy. And in those cases, every single kid who later developed autism had elevated acetaminophen in their cord blood. So
it's one of the things I recommend. Just people avoid. It's just avoid it. Absolutely. Hey, one thing on the organic acid test, right? We often see elevated B5, so pantothenic acid, and I'm wondering if that is, coming from active supplementation, or is there some type of conversion problem perhaps that's occurring in converting pantothenic acid into its active form? Because there's that pantothenic acid kinase 2 enzyme, which I know the severe genetic mutation of that's quite rare, but there's always variants that may be existing. What are your thoughts on that?
So pantothenic acid could be, as you point out, if the conversion is not going correctly, but it might also be because if the coenzyme A is not is all being used up by that might cause a blockade in the conversion. So I think it would be worthwhile to consider, in addition to obviously to treating the Clostridia, to avoiding the GMO foods, and I mean, it may be very worthwhile to consider supplementing with pantothenic acid as well. Coenzyme A itself is only available in tiny amounts from biochemical companies. So it's probably, and then even if it was taken, I don't know if it would be absorbed into the body. So I don't know if that's an option. Perhaps it might be, but and now it would be too expensive to be practical, because it's even it's hundreds of dollars for a few milligrams.
Would you see any advantage in something like pantothene, which is more of an active form, sort of like saying, using more of an active form?
Yeah. So all of these things I think are worth investigating, and are very likely to be non-toxic in any way.
One of the other things that I've thought about with the whole palmitic acid potential is, would you advise for people to avoid palm oil in cooking if they don't know what their palmitic acid levels are? Maybe not so much the general population, but in the autism population. If people are using coconut oil or palm oil because it has a high smoke point, would that be advantageous to avoid? Maybe, difficult to know. I know it's not been tested, but I'm just wondering your thoughts on that.
Well, the palm oil really is not toxic. It's just it is being excreted because of the coenzyme A deficiency. So I don't know if there's any evidence that it would be harmful. I mean, everything is worth investigating, but I would think that the main factor is the deficiency of free coenzyme A, and that resolving that problem, you'd be able, if you start measuring palmitic acid in the urine, you could find out if, various treatments, if they reduce the palmitic acid in the urine. And so you could try that. But I have a feeling that the main factor is the deficiency of the coenzyme A.
That's a great point. So any final thoughts? What other topics, other areas you're starting to look into and research? Where is this taking you?
I think that at this point, the main thing is getting the word out about how common this Clostridia thing, how dangerous the glyphosate, the Roundup, is, because of altering the microbiome. Not only in the gut, it's altering it in the human gut, but it's also altering in our animals, our food animals. Yeah. In Germany, they're finding cows who are eating the contaminated feed that's containing Roundup were dying of botulism, and even some of the farmers walking through the fields develop botulism. I know that we had one case where a child with high HPHPA was reported, or was following up, and found that he had died of botulism after a couple weeks after the test showing high HPHPA. So botulism. So one of the things scientifically would be developing a confirmation test to find out which species of Clostridia are most prevalent. We know the ones that can produce phenylpropionic acid, but we don't know specifically which ones are the most common in autism and in other neuropsychiatric diseases. I thought about doing that. The difficulty with botulism is it's been used as a terrorist weapon, and so it's very difficult to get cultures of Clostridia botulinum because of concern that it might fall into the hands of terrorists.
You don't want to end up on the terror watch list, Bill. So, well, hey, Bill, I really appreciate it. It's been great information. I'll make sure that people get access to a link for your recent article. Of course, we know the organic acid test is a fundamental test. It's been a fundamental test in my practice for, ever since I started doing it 25 years ago and learning from you. So just want to thank you so much for all of your contributions over these past many years and your continued desire to research and dig into the literature and try to find connections for these different types of complicated and complex disorders. So it's great, it's very noble. And so, you're to be congratulated for that.
Oh, thank you. I really appreciate all your efforts too, Kurt.
Well, thanks so much for
great colleague.
Yeah, I appreciate that. Well, thanks so much for joining us, Bill, and take care.
You've been listening to the Functional Medicine Doc Talk podcast with Dr. Kurt Woeller. For more information about this podcast, go to functionalmedicinedoctalk.com.